Hello everyone, welcome back to my channel. This is actually a continuation of the histology series of epithelium. So those who are watching it for the first time, please see the previous session so that you will get a continuity. So here we are moving on with the simple or unilaminar epithelium. So as the term implies, the epithelia, which is made up of a single layer of cells touching the basement membrane, you call it as simple epithelium or unilaminar epithelium. So that is the first point. It is a single layer of cells and all the cells rest on the basement membrane. Now the shape of the cell, how big it should be, how small it should be, everything is decided by the cell volume. So cell volume is depending upon the function of the cell. Like if you need little cytoplasm and if you have a very few organelles and if the metabolic activity of the cells are limited or at a low level, then the epithelium will be of the shape squamous or sometimes low cuboidal. So that is how you get the shape of the cell. It is depending upon the cell volume. So cell volume is actually decided by the amount of cytoplasm, the amount of organelles and the amount of metabolic activity. Okay, so if there is little cytoplasm, if there are or if only few organelles and if the metabolic activity is very low, you get almost flattened or a small or a low cuboidal shaped epithelium. Suppose if the cells are highly active and you need a large quantity of cytoplasm to be accommodated within the cell, and if you have numerous mitochondria, numerous endoplasmic reticulum, then you have to accommodate a larger volume, and in that case the epithelium will be tall cuboidal or columnar in shape. Now again the epithelium will, depending upon the special features, you will have cilia, then you will have sometimes microvilli on the surface, then sometimes you will be having secretory vacuoles within the cell, and you will be having sensory nerve endings with sensory features, etc. Now there is one entity known as myoepithelial cells. So what do you mean by myoepithelium? Epithelium, we know a continuous layer of cells. If there are contractile elements in it, like that of a muscle, myo means related to muscle or which are contractile in nature. So myoepithelial means the epithelial cells which are capable of contraction. And where will you get such type of epithelia? You will be seeing these type of myoepithelial cells in association with glands, so that when they contract, they will help in secreting out its secretion. So that they are known as myoepithelial cells. Now let's see one by one. Depending upon the shape of the epithelial cells, we will be classifying the unilaminar or simple epithelium into different types. The first one is simple squamous. Squamous means flattened or polygonal. So simple squamous means they are simple squamous epithelium means they are the epithelium where you get flattened or polygonal cells. Now another important feature of this epithelium is they have tessellated. Tessellated means complex interlocking borders. So if this is a cell, you can see in between the cell they are actually interlocking with each other. This type of interlocking is known as tessellated. Now the third one is nucleus is actually bulging into the overlying space. That means you can see that this is the upper view. If you look from above, you can see the cells as flattened cells, just like interlocking cells, which we have on our pavement pathways and all. So if you look from above, the cells are flattened and you can see the interlocking borders between the cells. But if you look from the side, you can see that the cytoplasm, the amount of cytoplasm is very less. So when you get a nucleus here, only at that region the cell membrane will be actually bulging in order to accommodate that nucleus, whereas the rest of the region it will be almost flattened so that it will be closely in contact with the basement membrane because of the less amount of cytoplasm. So it will appear like the nucleus is actually bulging onto the overlying space. So these are the two views. When you look from the side, this section is actually passing through the nucleus of the cell. This is another cell. If you take a section here again, you can see that this the side view will be like you have a flattened cell, and this, because of the presence of this nucleus, it is actually bulging onto the surface. So this will be the side view, and this will be the view which you will be drawing in your histology record. So you can see this is the basement membrane. This is the basement membrane, and this is the cell. And in the center you can see a bulge in order to accommodate this nucleus, and below you have the connective tissue. So squamous epithelium, you can easily remember, like, you please keep two flat papers, okay, two flat sheets of papers, and in the middle you just keep a lead ball or something, a small sphere. If you just keep in the middle and you approximate the two flat papers, how will it be looking like? Only at the region of that lead ball, it will be actually having a bump like thing projecting onto the surface. That is exactly how a squamous epithelium will look like. Now, where will you get this type of squamous epithelium? You can get it in the alveoli of lung, then you can get in the renal corpuscles, especially the outer capsule, then you will be getting in the renal tubules, especially the thin segment. So all these regions are examples where you get the simple squamous epithelium. Don't worry when you see these examples here. When you study in detail about the lung histology, then the renal histology, then it will be easy for you to correlate it with the function. For the time being, just remember, you will be getting simple squamous epithelium in the alveoli of lung, in the renal corpuscle, and in the renal tubule, especially the thin segment. Now, what do you mean by mesothelium? We have already mentioned in the previous session. Mesothelium is nothing but the epithelium lining the cavities like the pericardial cavity, the pleural cavity, and peritoneal cavity, and which are of mesodermal in origin. And what do you mean by endocardium? Again, it is a modified epithelium seen exclusively in the heart and the adjacent bigger vessels. So that is endocardium. So just if you hear endocardium, it is nothing but epithelium specialized for the heart. Then endothelium means, again, it is a squamous epithelium which you will be exclusively getting in the blood vessels. Okay, so that is endothelium. Now when you talk about the function of the squamous epithelium alone, simple squamous epithelium, the main function is rapid diffusion of gas and water. Gas, as we have already discussed, in the alveoli of lung, you get squamous epithelium. So there the main function is rapid diffusion of gases and rapid diffusion of water, as in cases of renal tubules and all. Now, it also helps in active transport because of the presence of endocytic vesicles, the vesicles which will actually capture something and will pass on to the cell or the underlying tissue. They are known as cells with endocytic vesicles. So it will act or it will help in the active transport. The third one is, we have already mentioned that you get tight junctions in between the cells. So how will these particles be transferred into the underlying tissue? So this is actually some of the questions which usually the students come up with, and that means they are actually listening to the class, because or else they won't have any doubt, because we have already mentioned that the cells are closely knit and they are having tight interlocking regions. So how this is made possible, rapid diffusion and all. So it is not through the space between the cells, but it is through the cells. All these transfer happens through the cells and not through the intercellular region. That is very, very important. So because of the tight junctions between the cells, the diffusion and everything happens through the cells. So these are some of the properties of the simple squamous epithelium. Then about cuboidal, columnar, pseudostratified, I will be doing it in the next session. So please keep watching and please share and subscribe. Thank you so much.